this wee little video, we are going over the urea cycle and its significance. Video, we are going over the urea cycle, all the enzymes involved, all the energy, because it actually takes energy, like it burns calories to run the urea cycle. And today we're going over the urea cycle. All we're going to focus on is just the actual biochemical mechanism of the urea cycle. For a little bit of context, we are running the urea cycle to detoxify nitrogenous waste. When we are breaking down proteins or burning amino acids for energy, some ammonia is released, and ammonia is neurotoxic. Like, it is literally poisonous for your neurons, poisonous for your brain tissues, and we must detoxify this nitrogenous waste. And the way we do that is with amino acids and the urea cycle. Your urea cycle will be elevated if you are breaking down more body proteins, if you are burning more amino acids for energy, or if you have a very high protein diet, you will be increasing the synthesis of urea cycle enzymes. If you have a high protein diet, you are doing this urea cycle just way more because there is more nitrogen entering your body that must be detoxified and dealt with. So let's just start with the urea cycle and what the frick is going on and where it is even taking place. The urea cycle is happening all the time, and it is happening in your liver, in the cytosol, and in the mitochondria. And under what context? Well, it is happening all the time because there always is some amount of nitrogenous waste being produced. There's always some nitrogen or ammonia that we need to detox as a result of normal metabolism, but especially under conditions of high muscle protein breakdown or high rates of amino acid oxidation, or in high protein diets, there will just be more nitrogenous waste that we need to deal with. So inside of your liver, we start with glutamine. Glutamine is an amino acid, and it is how your body transports safely nitrogen around, because we cannot just send ammonia or nitrogen straight into the blood. It needs to be transported in some safe, non-reactive manner, and we do that using glutamine. Glutamine shows up in your liver mitochondria, and glutamine is a nitrogen-containing amino acid. So glutamine is how we are transporting the nitrogen produced in your muscle cells as a result of amino acid breakdown to your liver, because we cannot just send ammonia out into your blood. That is toxic, it is neurotoxic, and it will cause a huge amount of cellular damage and damage your brain tissues, damage your neurons. It's just bad. We can't do that. So what we do is we use glutamine as a way to transfer that nitrogen around your body safely. Glutamine is kind of like a nitrogen escort. So glutamine escorts that nitrogen to your liver, and it enters your liver mitochondria. The first thing we do here is we use the enzyme glutaminase to remove the amine group, and then that gives us glutamate. So glutamine comes in, the enzyme glutaminase spits out ammonia, and it spits out glutamate. This ammonia, we still need to process it all the way around here to detoxify as urea. The first thing we need to do to actually get this ammonia into the urea cycle is we need to turn it into carbamoyl phosphate. This is a very energetically expensive reaction because carbamoyl phosphate has like a lot going on. First of all, we are adding a phosphate group to it. A phosphate is PO43-. There's a lot of negative charge and negative charge repulsion going on. So we know this reaction is unstable, unfavorable. It's probably going to be very expensive. We are also dealing with bicarbonate, so we have to stick this bicarbonate molecule onto the ammonia as well as this phosphate group. That's why it's so expensive. We use the enzyme carbamoyl phosphatase 1, CPTI, to facilitate this reaction. So zooming in, bicarbonate comes in, 2 ATPs come in, ammonia comes in. We spit out carbamoyl phosphate here, and then we spit out 2 ADP and 1 phosphate. So 2 ATP come in, 2 ADP go out, 1 phosphate goes out, and 1 phosphate is actually within this molecule because it's carbamoyl phosphate.
Once we yield this carbamoyl phosphate, we can now really start the urea cycle.
What we do now is we take ornithine. ornithine is a critical molecule intermediate in the urea cycle, and this ornithine enters the mitochondria.
Actually, let me just make this more clear.
Mitochondria up here, cytosol down here. Once we have carbamoyl phosphate, ornithine goes from the cytosol into the mitochondria, and ornithine hooks up with carbamoyl phosphate to spit out citrulline and a phosphate molecule. And this is done by the enzyme ornithine carboxylase. Yeah, that's what I wrote.
Sometimes when I'm writing so messy, I can't figure out what's going on.
But yes, this is the enzyme ornithine carboxylase, and we take ornithine, we hook it up with carbamoyl phosphate, and we spit out phosphate, and we make citrulline. Once we have citrulline, we can really start spinning the urea cycle. So citrulline goes out of the mitochondria and it goes into the cytosol, where it is acted upon by the enzyme argininosuccinate synthetase, and it is a synthetase, so we know it uses ATP.
Synthase, no ATP. Synthetase, yes, ATP is needed.
So citrulline in the cytosol is acted upon by argininosuccinate synthetase, where it uses 1 ATP to stick AMP onto citrulline. This gives us a citrulyl AMP intermediate, and it spits out PPI. Then the amino acid aspartate enters the cytosol, and we switch aspartate onto citrulyl AMP, and we use aspartate to kick off AMP. So we have citrulyl AMP, aspartate comes in, kicks off AMP, and now we have argininosuccinate. Argininosuccinate is then transformed into arginine, and in that process we actually make fumarate, and we use the enzyme argininosuccinase. Yeah, we use the enzyme argininosuccinase. So we have argininosuccinate, and then we spit out arginine and fumarate. If you are very familiar with the tricarboxylic acid cycle, you will recognize that fumarate is an intermediate in the tricarboxylic acid cycle. So this fumarate in the cytosol can be transformed into malate. Malate can enter the mitochondria, and then that malate can be fed back into the tricarboxylic acid cycle, and we can get a little bit of ATP from that.
Let's just stick to the urea cycle.
So fumarate is spat out, and we have arginine. Now we use the enzyme arginase to actually make urea. Arginine comes in, water, HOH, H2O, water comes in, arginine comes in, and we synthesize urea. Urea travels out of your liver to your kidneys and you pee it out. This is officially how you detoxify that nitrogenous waste. It is excreted in your urine as urea. Once we process arginine and water through arginase and we get rid of urea, we are back at ornithine.
And ornithine, what it does is it travels back into your liver mitochondria, and then it is hooked up with another molecule of carbamoyl phosphate, and it just keeps going around and around over and over and over again, because this is a cycle, and it is the urea cycle, so it is a cycle that just keeps on spitting out urea.
Let's focus on some of the more interesting specific details of the cycle. If you are on a high protein diet, you could potentially consider supplementing with ornithine, citrulline, or arginine, and that is because after running this cycle so many times, some of these molecules may break down, like they might experience a little bit of wear and tear or oxidation. If you are supplementing with ornithine, citrulline, or arginine, you could be helping out replenish the urea cycle intermediates. So you could just be helping this cycle operate at maximum efficiency and therefore help your body detoxify all this nitrogenous waste. Okay, this was everything I wanted to talk about in today's petite little video on the urea cycle. Let me know in the comments what you liked and what you learned and what you found was just like so exciting and interesting. I would love to get that feedback. And then also, please like, comment, and subscribe. It helps me so much put more time, energy, and resources into making these videos and making them better, because I know I can be a little all over the place sometimes. Okay, everybody, have such a rocking day, and I'll see you in the next production. Goodbye. Here are glutamine. It brings the amino,